In this short tutorial, we explain the basics of working with the InsideDNA platform. While it's very easy to work with our application, there are some nice tricks you may not know from the first sign-in. With this tutorial, we want to make sure you know all the tricks and can get the most of the InsideDNA functionality. First, create an account on the InsideDNA website, either by registering or using your Gmail or Facebook accounts, and log in. In the platform, you have three main tabs on the top navigation menu: Tools, Tasks, and Files. In Tools, you can search for bioinformatics tools, organize them into projects, and run. In Tasks, you can monitor progress of your submitted tasks and do some operations on them. In Files, you can manage all your data pretty much like any normal desktop file manager. There is a very powerful searching engine beside InsideDNA tool database, which makes it easy to discover not only packages, but also specific functions within software that suits your needs. To search for a tool, simply use Search field in My Tools and type any keyword that may be relevant to your query. The results can be presented in thumbnail view or in table view. Moreover, you can always subset your search results by specific keywords or keywords combinations. When you find a tool that looks interesting, you can obtain more information about the tool by pressing Read More button. It is very handy to organize tools by projects because it is then much easier to find which tools and with what settings you used, and your research becomes more reproducible. To create a new empty project, click on Add New Project and select a project name. You can always add tools to your projects. For example, type in search box Top Hat and then click on Plus button and choose Arabidopsis project in the drop-down list. To run a tool, you need to initialize Tool Settings menu by clicking on Run Tool on Tool thumbnail. Once you click on the Run tool, you will have a Tool Setting menu opened. Here, you need to specify the task's name, tool parameters, and how much core and CPU you need. Then you will need to preview the task and submit it. Specify the task name, which will make sense for you later on. Clicking on the Browse button will open a mini file manager that facilitates the choice of input and output files and folders. There, you can also create a new folder or upload data. It is a good idea to always preview the task before submission. This way, you can have a quick check of the specified parameters and also familiarize yourself with the command line way of doing bioinformatics. Next, you will need to select how much computing power you want for the task. It is a good idea to start with a smaller queue. For example, in our case, it will be sufficient to either launch task with 4 cores and 15 GB of RAM. Once you selected a compute capacity, simply click on Submit Task button. Once submitted, you can either go straight to the task monitoring option View Submitted Task or stay on the page. For now, click on the View Submitted Task. In Task Bookmark, you have several options. Tasks are grouped by their status. Typically, an error-free task first appears in the running group and then automatically moves to the completed group. If you submit more than 5 tasks at once, extra tasks will be put on hold pending group. Because right now, you can run simultaneously 5 tasks. Pending tasks will be launched as soon as one of the running tasks is completed. You can view error log, tool settings, and general parameters for each of your tasks. Moreover, you can always submit us a bug report, and we encourage you to do so if you have any issues. Now, let's move to the File Manager. Click on Files and navigate into your root directory. Here you will see all files, plus 2 extra folders with task errors and task logs. You can always preview what's inside the file by clicking on the Preview button on the right. Many more operations typical for a normal file manager are available: copying, moving, deleting, renaming files and folders, creating new folders, uploading and downloading files. Enjoy the InsideDNA and sequence crunching.